Very good evening, everyone. Please confirm if you all can hear me. Please write in the chat if I'm loud and clear and my screen is visible. So welcome to another session of Brahmastra Series. And in this session, I will be discussing questions, very important part C, CSIR NET questions from Unit 13, that is Methods in Biology. Thank you for confirming, Vijay Lakshmi. Very good evening. Thank you, Pulkit, for confirming. So how's your preparation going on? I hope you all are practicing and revising, practicing questions and revising the important concepts. Because today in this session, we will be discussing the questions based on some very important concepts. So let's get started and let's have a look at the very first question. So here's a question on your screen. The question is based on molecular cloning. The question is, given below are approximate lengths of DNA fragments obtained on agarose gel electrophoresis following restriction digestion of a 3 kb circular plasmid with different restriction enzymes. So as you can see here, using BAMH1, using HINC2, using EcoR1, and even on performing double digestion, we are getting different number of fragments and also different size fragments. Now, based on the above observation, that is based on the number of fragments and size of fragments, which one of the following statements is incorrect? And 4 statements are given to you. So I'm giving you some time to solve this question. Please solve it and let me know the answer, and then we'll discuss this question. Very good evening, Malini. Good evening to everyone. I have just started. I hope you all are excited for this session, for practicing questions, clearing your doubts. So these type of questions you will mostly find in part C of CSIR NET Life Science exam. And these questions are very scoring. If you know how to solve them, if you know the strategy of solving such questions, you can easily score 4 marks. So yes, let me see how many of you are able to answer this question. Pay attention to the details, what is given in the question to solve this question. So I'll just give you a hint. Based on information given in the question, try to redraw. Try to redraw the figure and then perform the digestion and see what size of fragments you are getting, and then analyze the statements given in the question. Okay, Malini, you have already answered. It's option 2. Okay, so I'll wait for others as well. Okay, some more minutes I'm giving you. Please keep a track of time as well. Okay, in the exam, you have to solve the question within the given time limit, right? So you don't have to spend too much time on a single question. Okay, option 3, yes, fine. See, you all have to choose the incorrect statement, not the correct one. So pay attention to the terms which are given in the question. Is it incorrect? Is it correct? It is true? It is not true? Okay. Okay, Pulkit, you are also saying option 2. Fine. Let's see what others, what is the answer of others. When you practice such questions, you'll gain confidence. Okay, when you practice such questions, first of all, you will understand whether the concepts you have gone through related to molecular cloning you are able to recall or not, you are able to understand the question or not. right? So practicing question is one of the very effective method of active learning, of revision also. We have to discuss lot of questions. So if you give me answer quickly, I will be able to cover all those questions because we have limited time. We have 1 hour session only. So please try to answer. I can see some more are joining. Please solve. Please try solving this question, everyone. Okay. So let me tell you the strategy of solving this question. The very first thing is you have to read the question carefully and you have to understand it, right? Now, based on the information given in the question, as you can see, the several restriction enzymes are mentioned and the fragments generated on performing restriction digestion with these enzymes, they are different. So you have to go through the details carefully and then try to redraw the diagram of the plasmid, 3 kb circular plasmid. So if we go through the details given, it is mentioned that the restriction digestion of 3 kb circular plasmid is performed. Now, you have to draw a restriction map over here of this circular plasmid. You have to determine the number of restriction site of these restriction enzymes given and also their location. So as you can see here, BAMH1 generates 2 fragments, whereas EcoR1 and HINC2, they generate only single fragment. So here you have to recall the concept that when we perform restriction digestion of a circular plasmid, the number of fragments generated are always equal to number of restriction sites present of that particular restriction enzyme. So here, BAMH1, when we are getting 2 fragments, it means that 2 restriction sites are present of BAMH1 and 1 each of HINC2 and EcoR1 is present in the plasmid. Now, question arises how to determine the location of BAMH1, HINC2 and EcoR1. So we'll go for double digestion. We'll look the double digestion. So if you see here on performing digestion with BAMH1 and HINC2 together, we are getting a fragment of 2.5 kb, which is similar to fragment generated by BAMH1 when only digestion with BAMH1 is done. And another 2 fragments are of size 0.2 kb and 0.3 kb. So if I add them, I'll get 0.2 plus 0.3 gives me 0.5 kb only. So this means that HINC2 site, because BAMH1, what is common, what is different? HINC2. So HINC2 site is present in between 0.5 kb site of BAMH1. So as I told you, 2 sites of BAMH1 are there. So let's say this is one site of BAMH1 and this is another site of BAMH1. And then EcoR1 will be present, HIND3, sorry, please correct, HIND3 will be present between these 2 sites. Now, they are not equal size fragment. One is 0.2, another is 0.3. So you will place it like this. So here you will have HINC2. So one will be 0.3, another will be 0.2 kb. And this totals 0.5. Then if we look at EcoR1, now we have to determine the location of EcoR1. So if we look EcoR1 and BAMH1, the 0.5 kb is similar to BAMH1 0.5 kb fragment. But what is different? 1 kb and 1.5. That is on performing double digestion with EcoR1 and BAMH1, we are getting one fragment 0.5 kb, which is common when we are performing restriction digestion with only BAMH1. But we are getting, when double digestion is performed, we are getting 1 kb and 1.5 kb. So if I add them, I'll get 2.5 kb, which is the fragment I get when I perform the restriction digestion with BAMH1 only. So that means that this EcoR1 site is present in this 2.5 kb region. So let's say we put over here the EcoR1 site. So one will be like one side you will be getting 1.5 kb fragment, another side you will be getting 1 kb fragment. Now you have to pay attention to the terms. Okay, you have to pay attention to the term whether incorrect statement is asked or correct statement is asked. So as in this question, incorrect statement is asked, so we have to go through each statement one by one. So let's go through each statement one by one. Okay. So this restriction map I have drawn over here. So basically, this is the arrangement. Okay, so this is 0.2, this is 0.3, and 1.5 and 1 kb, and you can see HINC2 site is present between two BAM-H1 site, and this is EcoR1 site. Now, statement 1 is HINC2 and EcoR1 have a single recognition site each in the plasmid. So if we look at this plasmid, the 3 kb plasmid, so this statement is correct, right? Because if we simply perform digestion with HINC2, this is a circular plasmid, it opens up and we'll get a 3 kb size fragment. Similarly, if you perform restriction digestion with the only EcoR1, it will cut over here and we'll get the plasmid will linearize and we'll get a 3 kb size fragment. So yes, HINC2 and EcoR1 have a single recognition site, and that too I have already told you that because individually on performing digestion with simply HINC2 and EcoR1, we are getting only one fragment, right? So number of restriction site, number of fragments generated in case of circular plasmid is equal to number of restriction sites present. Then HINC2 site is located between two BAM-H1 sites. This is absolutely correct. You can see that between two BAM-H1 sites, we have the HINC2 site which is present. So statement 2 is also correct. Then statement 3 is that distance between EcoR1 and BAM-H1 site is less than that between HINC2 and BAM-H1. Now this is incorrect because you can see the distance between the HINC2 and BAM-H1 is less, right? But EcoR1 and BAM-H1 site is more, not less. So this is very important point, okay? EcoR1 and BAM-H1 distance, if you see one side 1.5 kb, if you see it from here to here, it is 1 kb. So it is not less, it is more in comparison to distance between BAM-H1 and HINC2, which is 0.2 kb and 0.3 kb. Then fourth statement is, so statement 3 is incorrect and we have to find the incorrect statement. So yes, the answer is option 3. Let's check option 4 also, which is HINC2 is located closer to one BAM-H1 site than the other. This is absolutely correct because you can see here this HINC2 site to this BAM-H1, it is 0.2 kb. However, BAM-H1 to this HINC2, like HINC2 to this BAM-H1, it is 0.3 kb. So yes, they are not equal. It is located, the HINC2 is located closer to one BAM-H1 than another. So because incorrect statement has to be asked, is asked here, so answer is option 3. Now moving over to another question. So Malini, you are absolutely right. It's option 3, not option 2. And I hope it is clear to you, Vijay Lakshmi, why the answer will be option 3 and not option 2. Pulkit, I hope it is clear to you now. Now let's have a look at another question. Given below are some statements with blank spaces indicated by this dash. So 4 statements are given. So go through each statement and you have to find that which one of the following options has the correct sequence of terms that can be used to complete the above statements from A to D such that all statements become true. So this is very important point that you have to find the true statement. That is, all the statements become true, right? So go through this question and let me know the answer quickly. So I'm giving you 3 minutes. Quickly solve this question. And if you can answer it before that, then it's really great. Keep the track of time when you are solving a question. So 4 statements are given. At least if you are able to recall or are able to find out, or like by going through the statements, if you are able to understand that what keyword, what term should come here by looking at the options, you can easily answer this question. Okay, option 3, Malini. Let's check. We'll check in some time. Please try, everyone. It's for your own benefit, right? In the exam, these are the questions only which you have to solve, right? Okay, Anupam, option 3, Surendra, option 3. Okay, good. So far I'm getting a uniform answer. Akash, option 3. Focus on goal, option 3. Okay. Go through each statement one by one, and even you have to see the options as well, and then based on that you have to answer the question. So the statements given are not very difficult if you know the concepts. If you have studied about the Chargaff rule, if you have studied about the enzymes, polymerase enzymes, right, you can easily answer this question. And even if you are familiar with about the techniques, then also you can answer this question. So plasmid, statement A is a plasmid cloning vector digested with an enzyme with a single restriction site in the plasmid that generates 3' overhangs can be made blunt-ended using what? So one more minute, quickly answer the question, those who have not yet answered. Done, everyone? Okay, Vijay Lakshmi, option 3, fine. So I cannot give you too much time because as I told you, we have a limited time for this session, Brahma series session. And I want to discuss as many questions as possible with you all so that you can revise and practice questions during the session itself. So the very first statement, let's go through each statement one by one. So statement A is a plasmid cloning vector digested with an enzyme with a single restriction site in the plasmid that generates 3' overhangs. 3' overhangs can be made blunt-ended using which enzyme? So here you can see in the options it has been given tag polymerase, PFU polymerase, Moonbeam polymer nuclease, and reverse transcriptase. So first of all, I hope you know like the reverse transcriptase, tag polymerase is a very popular polymerase enzyme. So reverse transcriptase, it uses messenger RNA as a template and synthesizes a complementary strand that is cDNA. Whereas tag polymerase, it is used in PCR because it is a thermostable, it can resist high temperature. And most importantly here, it is about that which enzyme can digest, can generate, like can digest the 3' overhangs and can be made blunt-ended. So nuclease are the enzymes and Moonbeam nuclease is the enzyme which is basically perform this function. So when I say Moonbeam nuclease, it is only given in option 3 and directly, like you don't have to devote to, gain too much time on this question. Simply you can say that answer is option 3. But let's go through other statements as well, since we are understanding the question and try to understand the strategy of solving such question. So if we see statement B, DNA with a nucleotide composition of 30% A, 35% G, 20% C, and 50% T. So if you are familiar with Chargaff rule, you know that in double-stranded DNA, adenine should be equal to thymine, right? So here, if you see adenine is 30% only and thymine is 50%. So that means it is not double-stranded DNA because in the options you have been given double-stranded and single-stranded. So it cannot be double-stranded. And therefore, it is a single-stranded DNA as the amount of adenine is not equal to amount of thymine, and also the amount of guanine, which is 35%, is not equal to amount of cytosine, which is 20%. So again, this confirms our answer that B is single-stranded. Then statement C is that production of only truncated molecules of transgene derived messenger RNA in transgenic plants generated using a transgene. So here I would like to give you a concept of polyadenylation signals, because if you see the options in one, it is mentioned presence of messenger RNA instability sequences, codon usage variation, presence of polyadenylation signals in transgene sequence, and absence of poly A tail. So definitely, if you are looking this statement first, you should be thorough with what the concept of polyadenylation signal or messenger RNA, presence of messenger RNA instability, what it does, absence of poly A tail, and so on. So remember, polyadenylation signals play a crucial role in post-transcriptional processing of messenger RNA molecule in eukaryotic organism, including transgenic plants. And addition of poly A tail to 3' end of messenger RNA is important for messenger RNA stability, right? So if there is a presence of polyadenylation signal within a transgenic transgene, it can lead to production of truncated messenger RNA molecule. And therefore, option the C, the blank which is given in C should be presence of potential polyadenylation signal in transgenic sequence. And if we see the statement D, that is dash is a method for identifying the positions where individuals' DNA binding proteins attached to a genome. So again, it's a very direct statement. You have to just recall the technique which is used for identifying the positions where individual DNA binding proteins attach. So definitely it's not fluorescence in situ hybridization where we detect a particular DNA sequence in our sample. Here the interaction is there, DNA and protein, right? So this is only achieved using the chip sequencing, that is chromatin immunoprecipitation followed by sequencing, which is a technique widely used to study protein interaction of the proteins with the DNA. So interaction, it is used for probing protein DNA interactions. And PFG, pulse field gel electrophoresis, it is mainly being used to separate the large genome like of that of mammals. So again, this confirms our answer. That answer is chip sequencing, which is given in option 3. Then, so I'm glad to see that most, like all of you, those who have answered this question are able to answer it correctly. So very good, keep it up. Moving over to the another question, which is a quite tricky question. And again, it's a concept-based question. So please go through this question. The question is shown below are the circular dichroism spectra of a protein recorded under two different conditions, A and B. Now, from the options given below, select the one that is best interpretation of the spectra. So again, to answer this question, you have to go through each statement one by one. So kindly go through each statement and then again, I'm giving you some time and then I'll explain this question to you. So to answer this question, you should know about the application of circular dichroism spectroscopy. And you should know that which region of electromagnetic radiation is used to study secondary structure or tertiary structure. And also you should know how to read this spectrum, circular dichroism spectrum, which is given. So on x-axis wavelength is given and on y-axis ellipticity is given. So how will you answer this question? Go through the figure carefully given in the question. See, the advantage of such type of questions is that you don't have to think out of the box. These questions are very direct. The figure is given, so based on the figure given, you have to just analyze the figure, go through the statement, and based on that, you have to choose the, like here, best interpretation is asked, so you have to choose the best interpretation of the spectrum. Okay, already one of you have answered option 1. Okay, good. I'm glad to see that you all are able to answer the questions quickly. Very good. Read the question carefully, go through the figure, understand the figure given, and then correlate it with the statements given. And kindly note, like two questions we have discussed so far. So if you are not able to get it correct, if you are not able to answer it correctly, don't dishearten, okay? Because this is how you learn. And I'm sure that now once you have understood the concept, the strategy of solving the question, you will not commit the same mistake if the similar type of question occurs in the exam, right? So this is just for practice. Don't get disheartened if you're not able to answer it correctly, okay? This is how you learn, right? When you commit a mistake. But make sure that you don't commit the same mistake at the exam. So yes, quickly answer this question. You just have to recall about alpha helix, beta sheet structure, random coils, like how you will get to know that a protein contains alpha helix, beta sheet by looking at the peaks, by looking at the spectra. So there is a particular wavelength where on absorbing a particular electromagnetic radiation, the alpha helix show the absorbance peak. Similarly, beta sheet shows a peak. So actually here we get a not peak, we usually say positive band and negative band. So like here you can see the values above zero, it indicates the positive band, and the value below zero, that is gives the negative band. Here you will get the negative band. Okay, Hari, option 3, Vijayalakshmi, option 1. Fine. 1 more minute I'm giving to everyone. I can see everyone is not solving the question. Please try solving the questions. Done, everyone? Okay. So let me explain you the strategy of solving such question. So first of all, as you can see here, this question is about circular dichroism spectra, and a spectra is given. So first of all, you don't have to think out of the box. You have to analyze the spectra carefully, understand it carefully, and then go through the statements one by one. So in the like if you go through the statement is that protein has a helical secondary structure under condition A, which is denatured under condition B. Second statement is protein has a helical secondary structure under condition A that is converted to beta sheets under condition B. Third statement is spectra represents tertiary fold of the protein with condition A corresponding to mixed alpha helical plus beta sheet fold, and condition B corresponding to largely beta sheet fold. And the fourth statement is difference between the spectra under condition A and B is due to lower protein concentration under condition B. So basically, here you have to recall one concept. As I told you, alpha helix structure, beta sheet structure, random coil, they show a particular band at a particular wavelength. So first of all, since secondary structure, alpha helix and beta sheet are secondary structure of proteins which are mentioned in the question. So remember, far UV region, that is 190 to 250 nm, is the region which has been used to analyze the proteins if we want to study the secondary structure. So far UV circular dichroism provide information about kind and content of secondary structure. Remember, near UV region is used to study the tertiary structure of proteins. Now, since only secondary structure is mentioned, we'll focus on far UV. We'll use far UV region of far UV region, and we'll analyze the proteins. So the point that you have to recall here is that if alpha helix structure is present, remember that we'll get a two negative bands. One is around 208. So you can see on x-axis wavelength is given. So 208 is somewhere here because this is 210. So yes, we are getting a one band at 208 and another at around 222. So if such bands are given, negative bands, it indicates presence of alpha helix. Even a strong positive band around 192 nm indicates presence of alpha helix as shown over here. So the red ones, the red bands indicate the alpha helix here, and the blue one, the blue bands that you see over here, positive and negative, they are for beta sheet, and green one is for random unordered conformation. So how will you determine whether beta sheet structure is there? If beta sheet structure is there in your protein, you'll get a negative band centered around 216 to 218 nm. So we have to see the blue peak, and you can see it is centered around 216 to 218. So this indicates the presence of beta sheet. And also you'll get a positive band centered around 195 nm. So in the question, a spectra is given to us. So you just have to recall this concept and you have to just see whether the spectra given to you in the question show these peaks or not. So if you look here for A, you can see that here, like one is represented with blue dot line, another is represented with black dot line. So if we see the blue dot line, you will find, which is A, you will find that we are getting a positive band. This is positive, right? Positive band around 192. And also we are getting a negative bands over here, right? Two negative bands. One is centered around 208 and another is centered around 222. So this means what? Alpha helix is definitely present. So for protein in A condition, a strong positive band around 192 and two negative bands centered around 208 as well as 222 nm indicate presence of alpha helix structure. Now, if we see the black dotted line, you can see it becomes flat over here, right? So it means that the protein, the secondary structure protein of the protein is denatured, because as I told you, far UV region only detects the secondary structure, near UV detects the tertiary structure. So if you are getting a flat line after seeing a band up in another condition, when you change the condition, you're getting a flat line, it indicates protein is denatured. So if we look at the statements given, the options given, only the best interpretation which falls with the result is 1. Statement 1, that protein has helical secondary structure, right? And it is denatured. It is not converted to beta sheet, because if it is converted to beta sheet, we would have seen the band, right? Corresponding to beta sheet, right? Positive and negative band, which is not there. Similarly, again, we don't see the tertiary fold of alpha helical plus beta sheet. This is incorrect. And also, this is also incorrect. We have to choose the best interpretation. So according to the spectra given, answer is option 1. That is the best interpretation. So to learn the strategy of solving such Part C CSIR NET questions and to learn the concept, like if you have not started the CSIR NET exam preparation, or if you want to again revise your concepts, you can always join the batch. So Josh batch is the upcoming batch and the details are given on your screen. So you can see these are all the limited seats are available, and these are all the things you will be getting along with the Josh batch. So if you're interested, you can call us on our toll-free number, which is on your screen, or even you can visit stores.biotechnica.org for details. So our new batch is starting from 1st of July 2024. So answer of previous question is neither 3 nor 2. It is option 1 only. So yes, is it helpful? Are you enjoying solving questions? Please let me know in the chat. Are your concepts getting clear? Are you learning the strategy of solving Unit 13 Part C questions? Was it helpful so far? Okay, that's great. Thank you, Malini. Thank you, Shotel, Akash. Glad to see your response. Now let's move over to another question. Now this is a question, again a spectra based question. So this question is based on mass spectrometry. So see, in the exam, you may get question on UV visible spectroscopy, numerical question on Beer-Lambert's law, question on mass spectrometry, question on circular dichroism, or combination-based question where in the options different spectroscopy or mass spectrometry technique is mentioned. So don't do selective study when you are studying spectroscopy. That is your subunit C of Unit 13. It is about biophysical methods where you study all the spectroscopy techniques. So don't do selective study. At least learn the important ones, that is NMR, UV visible, NMR, circular dichroism, mass spectrometry. Okay. And be familiar with what happens in fluorescence spectroscopy and also infrared spectroscopy. So let's go through the question. The question is mass spectrum of a pure peptide recorded in the positive ion mode is shown below. So this is the spectrum. Now, 3 statements are given. What is the reason for multiple peaks in the mass spectrum of pure peptide? which peak corresponds to the monoisotopic species of the peptide, and what is the monoisotopic mass of the peptide. So based on analyzing this spectrum, mass spectrum, where on x-axis mass to charge ratio is given and on y-axis relative abundance is given, you have to select the right answers from the options given below. So again, I'm giving you some time. Please quickly go through this question and let me know the answer. So recall here the principle and application of mass spectrometry. Recall how you read the mass spectrum. Okay, Anupam, option 1, very quick, very good. So you can see in the options either carbon-13 isotope distribution or carbon-14. So if you see in two options carbon-13 isotope distribution and in two options carbon-14. So the best strategy to solve this question is omit two options out of four. If you are confused, omit two options out of four, and even you need to omit two options out of four. So when you are left with two options, then you can finally, like if you say it's carbon-13, then that means it will be 1 and option 3. So now you have to check for B peak and for C, for the B statement and C statement. Or if you say carbon-14, which is given in option 2 and 4, then again you have to check for B and C, whether it is peak 1 or peak 5, or it is the mass is 1124.55 or 1128.56 delta. Okay, Vidya Lakshmi, you are saying option 3, Malini option 1. Okay. Please try, everyone. Try solving this question. You don't have to devote separate time. And the advantage of Brahma series is that you can solve the questions in this session live, right? Like you don't have to devote extra time. This one session is all about question practicing. And even we can discuss live the explanation, and even if you have any doubts, you can ask me in the chat. Okay, Sony, you are saying screen is not clear. Sony, just try to check your settings. You have to check your setting to 1080. Okay, maybe you are not seeing at the highest pixel value, so you just have to check your settings, screen settings, because I'm also seeing on the screen. To me, it is absolutely clear. It's not blurred. So just check your settings once. High resolution, you have to keep it to high resolution. Okay, so Chandrima, option 3. Shortail, you are saying option 3. Okay, so majority says option 3 for this. Even option 1 is the answer, is being answered. So let's check. First of all, mass spectrometry is what? It's a technique which is using which we can determine it's an analytical technique that is used for analysis of sample. And using this technique, we can determine the composition of a sample. Like for example, if you are analyzing protein, you can analyze what all amino acids are present in that protein or a peptide, right? Even mass spectrometry has a wide application. It is used to study post-translational modifications. And also, we can use it for sequencing shorter polypeptides. Like we have MSMS spectrometry, tandem mass spectrometry, which is widely being used for sequencing shorter polypeptides. So it is asked in the question about A, B, and C statement. So A is, or A, B, and C part of the question is given. So A part is, what is the reason for multiple peaks in mass spectrum of pure peptides? So you can see we are getting like 5 peaks over here. And by looking at the options, we have got an idea that it is about the isotope, whether it is carbon 13 or carbon 14. So when we talk about carbon, when we talk about the carbon isotopes, what are isotopes? Those which are having the elements having the same atomic number but different mass number. Like when you talk about carbon, carbon has isotope carbon 12, carbon 13, carbon 14. They have the same atomic number, that is 6, but different mass number, that is 12, 13, and 14. Or to be more precise, they have different number of neutrons. So basically, when you talk about the reason for multiple peaks, it is related to presence of isotopes. And when you compare carbon 13 and carbon 14, see, carbon 12 and carbon 13 are stable isotopes of carbon. Carbon 14 is radioactive, right? So carbon 13, if you isotope, if you consider, it makes up 1.11% of all carbon atom. So that means approximately 1 in every 100 of these molecules will contain carbon 13. Carbon 14 is radioactive, so it decays to some other atom with time. So carbon 13, 1 out of 100 of these, 1 in every 100 of these molecules contain carbon 13 rather than more common carbon 12. So if we are considering carbon 13 in this CH4 group, carbon 13, so it means that 1 in every 100 of the molecule will have a mass of 17. How 17? Mass of carbon 13 plus mass of hydrogen 1. Excuse me, please. And since 4 hydrogens are there, so that total 17. And then if you consider carbon 12, so it is mass of carbon 12 is 12. And again hydrogen 1, so 4 hydrogens are there, so that is 16. So mass spectrum will therefore have a line corresponding to molecular ion both, and therefore you are getting multiple peaks here of carbon 13 also, carbon 12 also. Then, so basically, if you look at the options, when I say that A is carbon 13, that means answer cannot be option 2 and 4. So this way we have omitted 2 options out of 4. So that means answer will be between option 1 and 3. So what is B? Which peak corresponds to monoisotopic species of peptide? Remember, the peak corresponding to monoisotopic species of peptide is the peak with lowest mass to charge ratio. So if you see here, peak 1, if you see peak 1 to peak 5, peak 1 is having lowest mass to charge ratio. That is 1124.55. You can see it is increasing, 1125.55, 1126.55, and so on. So that means B is 1, peak 1, which is given in option 3, and therefore your answer is option 3. But we also let's study statement C, or the question C of the question C part of the question, that what is the monoisotopic mass of the peptide? Remember, monoisotopic mass of the molecule is sum of the accurate masses for most abundant isotope of each element present. So if you see the y-axis, it indicates relative abundance, and therefore height of the peak indicates relative abundance. So you can see peak 1 is most abundant, right? So it has a mass to charge 1124.55, and therefore we can say that the C is 1124.55, and therefore this confirms our answer. Answer is option 3, which is given by some of you. So answer is not option 1, it is option 3. Now let's move over to another question. These type of questions which you are seeing on your screen, this particular question, these questions are very scoring. Match the following type questions. Right? So again here, if you are familiar with at least like 4 techniques are given, if you are familiar with at least 2, you can easily answer such questions. So question is, given below are terms related to various techniques. Column X, in column X techniques are mentioned, and in column Y applications are mentioned. So which one of the following options represents all correct matches between column X and column Y? So I'm giving you some time to solve this question. Please go through it. The strategy of solving such question is that read the question carefully. Don't try to go through the entire table altogether. Okay. Go through the the technique which you are most confident about. Okay, so once you start with the most confident one, the technique in which you are most confident, the technique which you are sure, which you know the application, start with that, and then uh, go to another technique. So don't see the entire table altogether, and then look for the option which correctly represent the correct match between column X and column Y. So techniques given are mass spectrometry. Few minutes back only I told you about mass spectrometry technique, what this technique is all about. Then pulse field gel electrophoresis, if you remember, I told you what this technique is all about. And then we have isoelectric focusing and pack bio. Okay, option 2. Option 1, okay, Anupam, Malini, option 2. Good. Let's see the response of others. Always try to solve such type of questions in the exam because these questions are very scoring. Match the following type. Be it from any unit, try to go for match the following type questions first. Part C questions, they are very scoring. Okay, Sony option 2 you are saying? Fine, we'll check. Okay, Arzu, Vijayalakshmi option 1. Suchandrima, you are saying option 1. Okay, so majority is saying option 1. Okay, Shurthe, you are saying option 1. Done everyone? Or you need some more time? So recall about these techniques. Okay. So this is a easy question. You don't have to devote too much time on solving this question. I hope you are able to do it quickly. Comparative to comparison, comparing to other questions, I hope you are able to solve it much faster. So mass spectrometry technique is mentioned. So as I told you, it's a tool for detecting the composition for studying post-translation modification. So if you look at the application, it is mentioned identification of post-translation modifications in fourth option. So the fourth point. So that means A should be 4, which is given only in option 1 and 4. So that means answer cannot be option 2 and option 3. So let's say I am confident about mass spectrometry technique. So I start with that. If you're confident about pack bio, if you're confident about PFG, or if you're confident about mass spectrometry or isoelectric focusing, you can start with any one of the technique. Okay. Don't try to look at the table altogether. Then PFG, it is a pulse, stand for pulse field gel electrophoresis, and it is mainly used for genotyping, and it is used for separation of larger DNA molecule like that of mammalian uh, genome. So If we look at the application, so pulse field gel electrophoresis, separation of whole chromosome. So yes, this is appropriate. B is one. Then isoelectric focusing, it is related to isoelectric point, right? The pH at which this is the technique which is used for separation of the proteins, for separating different molecules like proteins on basis of difference in the isoelectric point. So yes, this is a technique which is used for separation of isoenzyme, and therefore C is 2. And PEG-bio is a sequencing technique. It's a single molecule real-time sequencing technique, smart technique, and it's a comes under third generation sequencing technology, and therefore D is 3. So if we look at the option, answer will be option 1. So A is 4, B is 1, C is 2, and D is 3, which is given in option 1, and therefore it is the answer. Now we have last 10 minutes of this session, so let's quickly solve 2 more questions. So here is a question which is based on gene knockout technology. The question is, a knock-in cassette was constructed in order to introduce the beta casein gene in an animal cell. The viable clones were selected using a double marker system with G418, which is a neomycin analog, and gencyclovir. You have to identify the correctly designed cassette from the options given below. So here are the 4 options, and you can see SOH is the sequence of homology, GOI is the gene of interest, NEO is for neomycin resistance, and TK is for thymidine kinase. Now again, this question is very scoring and easy. If you are familiar with the concept of how to create a gene knockout, uh, how to create a cassette to perform gene knockout, you can easily answer this question. So which is the correctly designed cassette from the options given below? So you can see the arrangement, like here neomycin is present inside single inside sequence of homology, and thymidine kinase is outside. But if you see option 2, thymidine kinase is also present between these sequence of homology region. In option 3, neomycin is present outside, and in option 4, both neomycin and thymidine kinase is placed outside. So what do you think, out of these 4 cassettes, which is the correctly designed cassette which results into the gene knockout or gene knock-in? Like this, the gene knock-in cassette, you want to introduce a gene of interest. Okay, Anupam, you are saying option 4. Fine. Yes, quickly give me the answer of this question. So you want to introduce beta casein gene. Okay, so the question can be on gene knockout or it can be on gene knock-in. So in gene knock-in, you want to introduce the beta casein gene in an animal cell. And the marker system used is G418, a neomycin analog, and gencyclovir. So what do you think is the correctly designed cassette here? Just recall what is the importance of sequence of homology, how the knock-in will happen, which strategy you will use. So just think about sequence of homology. What is the importance of sequence of homology? What does it allow? So this is your cassette, and let's say this is your the chromosome where you want to introduce your gene. So if I have to introduce the gene of interest over here, how I will introduce? I need to use some mechanism, right? Okay, Malini, option 1 you are saying. See, always remember when this cassette preparation is very much important, because to get the desired result, that is targeted gene insertion, this cassette is very important, because see, undesirable results can also happen. There is a possibility that this gene of interest is not inserted at the desired place. It is inserted randomly anywhere in the chromosome, or maybe no insertion happens. So these are undesirable results. Our desirable result is we want to introduce a gene of interest at a desired location at the targeted place in the chromosome, right? So remember, the cassette we have to draw is the cassette we have to construct is very important, and this is how your cassette should be. So actually, sequence of homology is needed for homologous recombination. The homologous recombination, which is a traditional method of performing gene knockout and gene knock-in, where we exchange the genetic material. In homologous recombination, exchange of genetic material happens between this sequence of homology, the region which is present, it has been exchanged. So you can see here, this is the cassette. If we are constructing, then onto the target, the homologous recombination will happen, and this gene of interest will be introduced at the right place, right? So as shown over here, we have the gene of interest along with neomycin resistant gene. In option 1, if you see, it is present in between. In option 2 also it is present. In option 3 it is present outside, and in option 4 also it is present outside. So always remember, ideally neomycin should be present in between the sequence of homology along with gene of interest. So what will happen when the desired recombination, the homologous recombination will happen, your neomycin resistant gene will also be introduced in your target chromosome, or at your target place along with the gene of interest. Now, if undesirable result is happening, undesirable result is where your gene of interest is not added at the targeted place. It is happening, it has been adding randomly anywhere in the genome. So remember, one more additional feature that you have to remember here is in case of homologous recombination, this thymidine kinase gene is lost. On the other hand, in case of random integration, the thymidine kinase is not lost. So how will you do the selection of desired and undesired result? We'll add the neomycin. So definitely those when your gene is inserted at the right place and randomly, both in that cases, the cells having these two kind of insertions, both will survive. So both will be resistant to neomycin. But when you add the ganciclovir, so thymidine kinase, it converts the ganciclovir into terminator of DNA synthesis, which results into killing of cell. So because in targeted gene insertion, thymidine kinase is lost, the ganciclovir will not result into killing, because it will not convert into the terminator of DNA synthesis. So in that case, the cells having the targeted vector recombines with the target gene, and targeted gene insertion is happening, they will be ganciclovir resistant, whereas where random insertion has happened, they will be ganciclovir sensitive. So ideally, if you see all the 4 options, the option 1 will shows you the most appropriate, most correct cassette. So I wanted to discuss some more questions. We hardly have 1 or 2 minutes left. So I would like to know if you have any questions for me, and anything that you want to ask, any questions you would like to ask. I hope these sessions are really helpful. Okay, I'm glad, Anupam, you like this question. So yes, this question was asked in 2023 paper. So I hope you are finding these sessions helpful. So every day, 6 to 7 p.m., we are having these sessions, and it will be up to the exam. We will be conducting these sessions so that you can quickly revise and recall the concepts and practice as many questions as possible. So any questions you have for me? Any suggestions? Anything would you like to mention? Yes, are you really enjoying these sessions? Was it helpful? Okay, helpful. I'm glad to see the response. Thank you, sir. Thank you so much. I'm glad you find it helpful, and I hope others also feels the same. So we'll see you in another session. Okay, Anupam and Malini, thank you for your response. I'm glad you find it helpful. So we'll come up with some more questions of some other unit in the upcoming sessions. So keep studying, keep learning. Thank you, everyone.